Terima kasih Cik Syam. Okey, sebelum kita ke bahagian 3 bersama Prof Madya Dr. Norrizah Jaafar Sidik, saya akan bacakan 2 soalan daripada pelajar. Okey, yang pertama Wan Muhammad Afif Nazrul Anwar. Okey, Doktor. Dia bertanyakan, Assalamualaikum Doktor, I am Wan Muhammad Afif from S1. I would like to ask a question. Why do we use term segments for hydrophilic and parts hydrophobic? Are they the same?
They are same, eh? They are same actually. The segments hydrophilic and parts, they are the same.
Okay, thank you Doktor. Okay, yang kedua daripada Muhammad Aiman. Okay, Assalamualaikum Doktor. Saya ingin penjelasan tentang how cell recognition involved in rejection by immune system. If there are no compatibility between blood type or of donor and recipient.
Okay, context of blood transfusion, the blood must match compatibility between the donor's blood and the recipient's blood, right? Okay, so this not always mean an identical blood match. Okay, so plasma transfusion, right? The red blood cell from a donor that is type, let's say O, can be transfused into patient of 4 different blood types, right? A, B, and O. Okay, so people with type O blood are called universal donors because they are donated red blood cells, have no A, B, or Rh antigens, right? And can therefore be safely given to people of any blood group. Okay, so people with type AB blood are universal recipients, because they have no antibodies to AB or Rh in their blood and can receive a red blood cell from a donor of any blood type. Okay, so plasma transfusion are meshed to avoid AB antibodies in the transfused plasma that will attack the recipient's red blood cell. So people with type AB blood are universal plasma donors. Their plasma does not contain A or B antibodies and can be transfused safely to all blood types. Okay, so here how cell recognition evolve. So because they have the site of they have the antibodies present and also antigen present, right? So they must match among this the cell, the cell must the antigen must match to the antibodies. Okay, that's all.
Okey, terima kasih Doktor. Okay, sebelum kita ada satu lagi soalan daripada Izrina Izlin. Assalamualaikum Doctor, I'm Izrina. As we know, cholesterol act as a fluidity buffer, but I read some other notes state cholesterol act as a temperature buffer. Is it considered the same?
Yeah, it's also act as a temperature buffer. Yeah, it can consider the same one, right?
Okay, thank you Doktor. Okay, kita boleh sambung ke bahagian 3 atau bahagian terakhir untuk chapter plasma membrane and transport. Okay, silakan Doktor.
Okay, I will continue with the lectures on the plasma membrane transport. So this slides, it shows the membrane structure result in selective permeability. So if you look at these figures, right? These are the phospholipid bilayer, right? So here shows the hydrophobic molecules such as the oxygen, carbon dioxide, and nitrogen gas, right? It can diffuse, it can dissolve, right, in the lipid bilayer. It can pass through the membrane rapidly, okay? Whereas there are other ions such as the chloride, sodium, potassium, okay, and also the small uncharged polar molecules such as the water and ethanol, and the large uncharged polar glucose such as the glucose and the sucrose. Okay, they cannot across the membrane easily. Yeah, they cannot across the membrane easily because of the hydrophobic core of the membrane. Okay, now we look at the transport protein where it allows the passage of hydrophilic substance across the membrane. So these channel proteins have a hydrophilic tunnel, okay, that certain molecules or ion can use as a tunnel, okay, and this channel protein we call it as aquaporins, okay, where it facilitates the passage of water, okay, only the water, okay, for aquaporin. The words aqua, okay, refers to water, aquapore. Porins come from the word pores, yeah, aquaporins. And then we have the carrier proteins where it binds to a molecule and change the shape to shuttle them across the membrane. So this one, it makes the ATP, the energy. Okay? So a transport protein is a specific for a substance it moves. Okay. Then we have this two types of transport across the plasma membrane. One is the passive transport, the other one is the active transport. Passive transport, it doesn't need any ATP, okay, or energy, whereas active, it needs the ATP. Okay, so passive transport can there are diffusion and the facilitated diffusion. Okay, so we will go for the different types of passive transport. Okay, the simple diffusion, for example, like the osmosis, and the facilitated diffusion. Okay, for simple diffusion, this molecule has tendency to spread out evenly into the available space. Okay, and the molecule will move randomly, but diffusion of a population of molecule may be directional. Okay, so molecules at dynamic equilibrium, as many molecules cross the membrane in one direction as in the other. So like the molecules, right, in given here, yeah, the example here, okay, you can see the dye here, the molecule of dye, it will cross, yeah, through the membrane, okay, until it will reach at the equilibrium level. Yeah, so the molecules diffuse from more concentrated, yeah, more concentrated to less concentrated. So meaning that diffuse down its concentration gradient. So if it is down, then from concentrated to less concentrated, okay, and leads to dynamic equilibrium. The solid molecule continue to cross the membrane but at equal rates in both directions. Okay, now we look at this this one, the next one. Okay, a simple rule of diffusion. In the absence of other forces, okay, a substance will diffuse from where it more concentrated to less concentrated. Yeah, a substance will diffuse down concentration gradient. Okay, no energy needed and a spontaneous process. Okay, a diffusion of water across a selectively permeable membrane from the region of lower solid concentration to a region of higher solid concentration until the solid concentration of both sides of the membrane are equal is called as osmosis. Yeah, osmosis. So osmosis is the movement of water through a semi-permeable membrane, according to the concentration gradient of water across the membrane, which is inversely proportional to the concentration of solutes. While diffusion transports material across membrane and within cell, osmosis transports only the water, water across a membrane, and the membrane limits the diffusion of solutes in the water. Okay, so you can see here, these are in the figures here, these are the green molecule here. The green color molecules is the sugar, right? And this part is the lower concentration of solute, and here is the higher concentration of solute. So the water will move through the permeable membrane, selective permeable membrane, okay, until it reach at the similar concentration of solute. So these are the U tube glass with a selectively permeable membranes where it's separating the two sugar solutions. So here water move from an area of higher to free water concentration, lower to higher solute concentrations. All right. Okay, so water molecule can pass through the pores, but sugar molecules cannot. Okay? Only the water molecules. Okay, so this side has fewer solid molecules, more free water molecules. Okay, whereas in this side, the water molecule cluster, you can see here, it will cluster around the sugar molecules, and this side has more solid molecules, fewer free water molecules. So, and it has the selectively permeable membrane at the center here. Okay? So this the osmosis. Osmosis. Only the water molecules will move. Okay, now we look at the tonicity. Okay, so tonicity can be described how an extracellular solution can change the volume of a cell by affecting the osmosis, or tonicity is the ability of a solution to cause a cell to gain or lose water. Okay. There are three types of solution. One is the isotonic, the other one is hypertonic, and the third one is hypotonic. So if it is isotonic, the solid concentration is the same. Is that inside the cell, and there is no water movement across the plasma membrane. If it is hypertonic, the solid concentration is greater than inside the cells. The cell will loses the water. Hypotonic, solid concentration is less than that inside the cell. Okay, so the cell will gain more water. So we look at the blood cell here. Okay, when place the blood cells in a solution that is hypotonic to the cell. So what happens? The water will move faster. It will enter the cell. Then it leaves the cells. So later the cell will swell and lyse. It will burst, because the water move enter the cell faster. For the isotonic, if the blood cell is immersed in the environment of the isotonic, there is no net movement of water across the plasma membrane. So the water flows across the membrane at the same rate, both direction, in and out. Yes. However, when put it in the hypertonic, the cell will lose water to the environment. Okay, and the cell become shrivel. It will become shrivel, and then it will die. So shrivel here, it will contract and then it will die. Yeah? Then this effect, okay, example given here is the effect of increasing the salinity of a lake to the animal. Okay, now we look at what happens when we put the plant cell in a place the plant cell in a hypotonic solution. Yeah? So remember that the plant cell have the cell wall. So when put in the hypotonic solution, the water enters the cell by osmosis. Yeah? The plant cell swells, but it does not burst. Yeah? The cell become turgid, become very firm. Yeah? And this is very important for the herbaceous plants. Yeah? So, and then we look at the isotonic solutions. When we put a plant cell, there is no net movement of water molecules. Yeah? No change in the cell volume, but the cell become flaccid. Yeah? And then it will wilted. Yeah? It will wits. And for hypertonic solution, yeah, the cell will plasmolyze. Yeah, plasmolyze. Okay, there's plasmolysis. There's process of plasmolysis occur here. There's a contraction of the protoplast of because loss of the water, the protoplast of the plant cell as a result of loss of water from the cell. Okay. So there is a net outflow of the water by osmosis from the cell. Yeah? The cell vacuole will shrink. Yeah? As you know that plants have a large vacuole. Yeah? So if there more water flows out, then the plasma membrane will push away from the cell wall. Yeah? And the cells will shrivels. All right. Okay, the movement of solid across the membrane will, with the help of transport proteins, facilitated. This is about the facilitated diffusion. Yeah. It follows the concentration gradients, increase the rate of diffusion across the cell membrane. Transport protein is very specific on which chemical they allow to pass through. Okay, so you can see here the transport proteins. Okay, there are channel here, right? Channel. The solutes can move through the channels, proteins here, and the carrier protein here, right? It will change the shape of the carrier proteins. Okay, channel proteins. Passageway that allows specific molecule or ion to cross the membrane. Okay, and hydrophilic passageway that allows water molecule or small ions to flow very quickly from one side of the membrane to another. So example like the aquaporins or ion channels. Yeah. So these are the aquaporins. Okay, you can see here the water molecules. Okay, aquaporins are the water channel proteins. It facilitates the massive amounts of diffusion of water molecules, yeah, in the plant and also in the animal cells. Okay, so if there is no aquaporin, the rate of water movement across the phospholipid bilayer is very slow. Okay. Now we look at the ion channels. It can be leaked channels or it can be gated channels. So what's the difference among these two? Between these two, leaked channels is always open. Yeah, it's always open. Whereas gated channels, it can open, it can close, okay, in response to a stimulus. This stimulus can be a chemical or ligand, yeah, or it can be electrical or change in voltage or mechanical. Okay. So now we look at the the ion channels here, the figures, okay, where as I mentioned just now, non-gated or leak channel always open. Okay, it's always open and and specific for one type of ion, although not absolute. Gated ion channels, yeah, the ligand-gated, it is an open or closed in response to ligand binding to receptor as acetylcholine. Okay, so the ligand-gated ion channels, permeability is greatly increased when some type of chemical ligands band to the protein structure. Okay, so a large subset function as neurotransmitters. So acetylcholine is a neurotransmitter. Yeah, as neurotransmitter. Can see here the receptor. Yeah, the receptor here at the phospholipid membrane, plasma membrane. Okay, so the receptor here, neurotransmitter, yeah, chemical, it will attach to receptor. Neurotransmitter chemical, it's like the acetylcholine, can attach to the bind site here, and the sites, the sites here, and then it will open. Just now, okay, without, if there is no neurotransmitter chemical attached before this, it's not attached to the receptor. It closed. Yeah? The gate is closed, right? But if there is a neurotransmitter chemical attached to the receptor, the gate will open. So the potassium and the chloride, the potassium and the sodium can move in and out through this these channels. Yeah, so this is the chemically gated ion channel. Yeah, so if you look the acetylcholine, yeah, the acetylcholine is an organic chemical that functions in brain and body of many types of animal, including humans, as a neurotransmitter, a chemical message released by the nerve cells to send signals to other cells such as neurons, muscle cells, and the gland cells. Neurotransmitter is the chemical that motor neurons of the nervous system release in order to reactivate or to activate the muscles. Okay, now we look at the voltage gated. This one, voltage gated, is open or closed in response to small voltage changes. Yeah, so voltage gated ion channels, yeah, known as voltage dependent ion channel also, are channels whose permeability is influenced by the membrane potential. Okay, so you can see here the positive charge and the negative charge. There's a membrane voltage changes occur here, right? So here, when the ions, yeah, when the ions cannot move in, okay, but if, because the gate is closed, but if there is a voltage changes, yeah, so this one is positive outside here, okay, and negative, but voltage change, the outside become negative, inside is positive, then the gate will open. This allow the ions to move in. Okay, so the voltage gated ion channel, you can see when it closed and when it's open. Yeah, so it open or close in response to the small voltage changes. Okay, now we look at the channel protein. Example given here is the cystic fibrosis. This cystic fibrosis is a kind of genetic disease, okay, where the mucus will block the airways. Okay, so it is characterized by the production of abnormally thick, sticky mucus, mostly affecting the respiratory airways. So mutation, the mutation can cause defectivity of the chloride channel, and chloride transport across the membrane closely linked to the sodium transport. Cell lining the respiratory tract cannot absorb the sodium chloride. So the natrium chloride will accumulate, affecting natural defensive protein, which is called as defensive, that kill bacteria in the lung. So bacteria triggers airways or cell lining the respiratory tract to produce excess abnormal thick and sticky mucus. So there's a clogged airways. This will damage the lung, and there's a failure of respiration. So you can see here the mucus blocks the air sacs. These are the alveoli in the lungs. The mucus blocks the air sac, and also the mucus blocks the airways of the respiratory system. Okay, carrier proteins. Okay, now we look at the carrier proteins here. Some small hydrophilic organic molecules, example like the glucose and amino acid, use carrier protein to pass through the cell membrane. Okay, by facilitated diffusion. Through it change the shape. The change in shape can be triggered by the binding of the solute to the binding site. So these are the binding site of the carrier protein, and release the transported molecule with no energy input is required. Okay, so these are the example. The glucose transporter, glucose bind to binding site of specific carrier protein, and the protein will change the shape, and then the glucose will be released into the cells. Okay, now we look at the example here. Okay, the carrier proteins, and the carrier proteins, cystinuria. Cystinuria, it is an inherited disease. This is caused by too much of amino acid cysteine. Also lysine or arginine in the urine. Normally, after entering the kidney, cysteine dissolves and goes back into the bloodstream. However, in people with cystinuria, carrier proteins that transport cysteine across the membrane of kidney cell are defective or missing. So there is no carrier protein. So as a result, cysteine accumulates in the urine and form crystal or stones. So it will block the kidney ureters or bladders. So you can see here in the kidney here, can see stones. Stones. So the cysteine accumulates in the urine, forms crystal or stones. You can see here the stones at the bladder, and also this part of the kidneys. So this is my last slide on the questions. So there are 3 questions. The first one is describe simple diffusion. Second one is what happened to an animal cell when immersed in the following solution, in the hypotonic and the hypotonic. And then name 2 types of ion channels. This is the last slide. With this, I end my lectures. Thank you.
Okey, terima kasih banyak-banyak Dr. Okay, juga terima kasih saya ucapkan kepada Profesor Madya Dr. Norrizah kerana sudi meluangkan masa bersama kami berkongsi ilmu dan pengalaman kepada pelajar sains asasi dengkil dan juga kami pensyarah biologi. Okay, semoga kita dapat berjumpa lagi di masa akan datang. Okay, thank you very much Doktor.
Okay, welcome. Jumpa lagi. Okay, terima kasih banyak-banyak.